My name is Anuradha Khade, and today the topic which we are going to see is from module number 3501, Genetic Engineering and Recombinant DNA Technology. And the topic which we are going to see is DNA modifying enzymes. Now, this DNA modifying enzymes, before actually going to the topic, let us first see what are the outcomes of this course, and basically at the end of this course we should be able to answer how we are going to associate all these different enzymes to use in the recombinant DNA technology, that is with the DNA. Now, before actually going to the topic, let us see some basic part of this topic. When we are talking about this modifying enzymes, basically we are going to utilize it on the DNA molecules. So prior going to this, we have some idea about the DNA structure, right? And this DNA structure is a double-stranded DNA molecule having two strands in anti-parallel direction, and all are having a basic structure, that is different types of four different nucleotides arranged in a sequence, and all these structures, this is having a direction. This is from 5 prime to 3 prime and from 5 prime to 3 prime. Every macromolecule in your DNA, in your cell, is having a specific direction. Everything is organized in our body. So I think if our DNA is, our genetic system is so organized, so we should also be in very organized system. So this DNA basically is a polymer of nucleotides, and each nucleotide is joined to one another by a phosphodiester bond, and each strand, this is one particular strand, each strand is bind to another strand which is also a polymer of nucleotides by a bond called as hydrogen bonds. Now all we know that hydrogen bond are the most weaker type of bond, although it is weaker, but n number of hydrogen bonds along the length of the DNA is adding the stability to the DNA molecule. Now why we all discussing this DNA structure is because these modifying enzymes have something to do with all these different types of bonds, and that is why we are seeing this DNA structure, basic structure, right? Now coming to the DNA modifying enzymes. So very first and bigger class of these DNA modifying enzymes is restriction endonuclease. Second class is DNA ligase. Third is terminal deoxynucleotidyl transferases. Fourth is kinases, and fifth is phosphatases. Now coming to the very first and the most biggest class of this restriction enzymes, all these restriction enzymes, they are found in bacteria. Now what is the role of these restriction enzymes when they are present in the bacteria? Usually all these restriction enzymes, when they are present in the bacterial cell, their role is, as we all human beings have immunity, bacteria have this enzymatic immunity. What they do is, this is a bacterial cell, this is the genetic material of the bacterium, and when the foreign particle, this foreign DNA enters into the bacterial cell in a natural condition, these restriction enzymes, which is the immune system of this bacteria, it kills, it fragments this DNA molecule and protects the existence of this whole bacterial cell. Now, this property of restriction enzyme, the cutting property of this restriction enzyme, we are exploiting this property into the recombinant DNA technology. Now why we need these properties is because in genetic engineering experiments we are interested in a particular gene or a segment of DNA which are of our interest, which may produce sometimes some enzyme or some RNA molecule or some which is giving us a product. And that product we have to identify from the whole DNA molecule. So in order to cut that specific sequence of the DNA molecule, we require this restriction endonucleases.